Hello, everyone. This is Mr. Schultz, and this is the FRQ review for AP Biology, specifically looking at cell energy FRQs. Now, before we start on the FRQ section, there are a couple of things I want to review for the actual AP test. So, we've seen this multiple times before, so maybe you want to skip ahead if you remember what this is all about. But the AP Bio test is only 2 questions long this time. It's 45 minutes or so. The first question is going to be a 25-minute question in which it's a free response question, specifically free response question number 1, which is interpreting and evaluating experimental results. It's generally an 8 to 10 point question that presents students, or presents you guys basically, with an authentic scenario accompanied by data in a table or graph. This question assesses student ability to do the following in 4 question parts. Part A is going to ask you to describe and explain biological concepts, processes, or models. So this is going to be like, hey, how does this specific thing work? So if we're talking about photosynthesis or respiration, it might be the whole process, or more likely, it's probably going to be one part of the process, like, hey, explain what happens during glycolysis. Part B is going to ask you to identify an experimental design procedure. So it's going to ask you, hey, what's the independent or dependent variable? What's a control? Something like that. It might also ask you to define part of that. So like, what does it mean to be a control versus what does it mean to be a constant? Part C is going to ask you to analyze the data, in which case you're going to actually look at the graph or data table to figure out what it is that you are actually looking at and interpreting it. And then part D is going to ask you to make and justify a prediction. So perhaps it's going to ask you to extrapolate the data, go beyond the data, or interpolate the data, which means look in between data points. And then it's going to ask you to justify that prediction using content knowledge. Okay? So it's highly recommended that for each section of the AP course, you make a cheat sheet or 2 per unit. So an example of a cheat sheet might be you might have 1 sheet on just photosynthesis and 1 on respiration. It just gives you a basic rundown of everything you need to know, specifically in terms of just content, okay? So it might have like a drawing of the general reaction. It might have other things that are gonna help you remember on the AP test. You don't wanna have a lot of these because if you end up rifling through them, you're gonna waste a lot of your time. So 1 or 2 per unit is an ideal number because then you can actually be like, oh yeah, this is on this sheet and I can go right to it. So keep that in mind. For the second question, you're gonna have 5 minutes in between to submit the first question. And then the second question is only 15 minutes. And this one's gonna be worth less points. It'll be just 4 points. It's gonna present you with an authentic scenario describing a biological phenomenon with a disruption. So it's gonna be something that happens in nature with some sort of thing that's gonna mess it up. Okay, they're gonna change something or remove something. And then this question is going to ask you to describe the biological concepts or processes. So again, that review sheet's going to be really helpful if you have the information written down already. Part B is going to ask you to explain the biological concept. Again, if you have really good cheat sheets that are easy to read and aren't too complicated or like full of stuff, it should be right there. But part C is going to ask you to predict the cause or the effect of a change in the biological system. So in photosynthesis, it could be like, hey, what happens if NADPH can't accept electrons? Or what happens if the chlorophyll can't give up an electron? So that might be something that happens. And then it would ask you to justify your predictions by explaining, hey, this is probably what would happen afterwards. In this case, if NADPH can't accept an electron, it can't use that later in the Krebs cycle to actually make G3P, which is going to cause the plant to not make nearly as much sugars. So keep that in mind. This is the actual setup of the AP test. Now, the other thing I want to mention, and I've probably posted it a few times via email and on Google Classroom, is that College Board actually provides you with a whole bunch of review videos, and they drop them every couple of days. In the next couple of weeks, you're going to start seeing more about like how to take the test and some general information about how to take and do FRQs properly. So besides the videos that I'm providing, there's lots of other information out there for the test. You can also expect in the next day or 2, I might also drop a practice AP test that'll be similar to the one that you would see coming up at the end or the middle of May, which the exam is May 18th at 1 p.m. Okay, so looking at cell energy FRQs. I'm going to zoom in a little bit so we can see the questions a little easier. Now, FRQ1 is simply an informational FRQ, which means that it will not show up on the AP test. On the AP test in and of itself, they might ask a question like this originally, which is like, hey, in this case, many biological structures are composed of smaller units assembled into more complex structures having functions based on their structural organization. For the following complex structures, describe the smaller units, their assembly into the larger structures, and one major function of these larger organized structures. So in this case, you're looking at, it says describe the smaller units, but it also wants to know how they turn into the bigger units and then their function. In this case, this is just a 4-point question where you need to describe the smaller subunits. In this case, the smaller subunits of the inner membrane of the mitochondria are phospholipids, because it's a membrane, and proteins, because it's a membrane that's got a whole bunch of electron transport chains and DNA or ATP synthase, okay? And then these would make up the inner membrane of the mitochondria, which is many, many folds to increase surface area, which allows for an increase in the amount of ATP made. So basically, for that question, they're just looking for a bunch of points. Specifically, it's more or less your understanding of one major function in biology. Again, not going to show up on the AP test. FRQ2, which is actually just the first two parts of one FRQ, would be something similar to what you could expect on an AP test this year. So a researcher is studying patterns of gene expression in mice. The researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of mRNA from 6 genes. So we're looking at 6 different genes and their mRNA. So this is a little bit of transcription and translation too. The data are shown in figure 1, which is right here. Based on the data provided, identify the gene that is most likely to encode a protein that is an essential component of glycolysis. Provide reasoning to support your identification. Now, for part A, it says identify, which means you need to identify specifically which of the 6 genes do you think is most likely to be it. And then you need to provide reasoning. You need to explain why. For part A, incomplete sentences, and you can actually bullet them like this so that way the grader can read them easier. But you can't use just bullets like short phrases and stuff like that. They have to be complete sentences. The gene that is most likely to encode a protein that is essential for glycolysis. So the gene that probably encodes for the essential protein is gene, and then we have to look at our graph. So this is really similar to that, hey, let's interpret some data. And so if we look, we'll notice that in the liver, heart, and brain, we find E, but not in the kidneys, pancreas, or muscles, skeletal muscles, and we just kind of scan across, and we notice that, whoa, J isn't found in any of these things. But G is found in all of them. And the word essential means that it's needed. So in this case, we would expect that the gene that probably encodes for the essential, the gene that probably encodes for the essential protein is gene G. The reason for this is because gene G is found in all the different tissues. So that's part A. Now part B is actually a transcription and translation and gene expression question. In this case, the researchers observed that the tissues with a high level of gene H, so we're up here, did not always have gene H protein. Provide reasoning to explain how tissues with high gene H mRNA levels can have no gene H protein. There's a couple of options for this answer. One of them could be that just because there's the mRNA for gene H doesn't necessarily mean that the protein H is there. This could be, one, a case of the mRNA is then being cut and snipped via like removing introns or exons to create a different version of a different protein that's similar to protein H, but not protein H. It could also mention, so you could mention like introns, exons being removed. Or it could also be that protein H, or the H protein, is being used to build maybe a larger protein complex, or it could also be an intermediary step in some sort of biological reaction. So understand that just because it's there, the mRNA is there, doesn't necessarily mean that the protein will be there. It could be the mRNA is then modified and then turned into a different protein. The H protein could be used in something else as an intermediary, or you could also say that there's some block on translation of the actual H protein. A positive or negative feedback loop, probably a negative feedback loop, is blocking gene H from actually being translated into the H protein. So this is FRQ2. And the first part, and actually the second part, are really similar to what you could expect on an actual AP test, particularly this year, because this first part is 2 points. And it could be like an 8 to 10 point question where they actually ask you about like, hey, these genes code for something. They might ask you to explain structure to function. What do tissues mean? What are they? What would you expect to find them? How do they work? Things like that. So just be aware that this part of this question is similar to what could show up on your actual AP test. Now, for FRQ3, again, this is one of those, hey, do you know a whole bunch of information about this one particular thing? And we don't really need to worry about this FRQ in terms of doing it for review for the test. However, it is a great review of the actual content itself, in which you discuss the role of green plants in transforming the sun's energy into a form that can ultimately be used by heterotrophs. So you need to explain, hey, how does the sun's energy end up in organisms that can't harness it? So in our case, we have to eat sugars to get it. And so you need to explain how plants get the sunlight into sugars, and then we eat the sugars, basically. So in this problem, they're going to look for very specific things. Specifically, they're going to look for a description of photosynthesis that includes chlorophyll, because chlorophyll is the big chemical at work here and what it does. Chlorophyll takes in sunlight, and then it's able to give up electrons to harness for making ATP or for NADPH to send to the Calvin cycle, which can then help energize and turn sugar or carbon dioxide into sugars. So there's just a whole bunch of stuff. And if you check the answer key, you'll be able to see that there's a whole bunch of things to answer this question. FRQ4 is very, very similar to what you could expect on that, hey, here's an experiment, show me what it means. In experiment, or FRQ4, an agricultural biologist was evaluating 2 newly developed varieties of wheat as potential crops. In an experiment, seedlings were germinated on moist paper towels at 20 degrees Celsius for 48 hours. Oxygen consumption of the 2-day-old seedlings was measured at different temperatures. The data are shown in the graph below. Now, before we even look at the question, we can kind of guess what we're going to be talking about initially, and that's oxygen consumption is talking about metabolism. Okay, we're talking about respiration. So even though they're seedlings, we're not going to worry about sunlight right now in photosynthesis. So part A asks us to explain the relationship between metabolism and oxygen consumption. And then it asks us to discuss the effect of temperature on metabolism for each variety of seedlings. So for part A, this is a 2-part question. And so on your FRQ sheet, and I apologize, I had to split it into 2 pages, I've got my question, and then I have to answer it. In this case, for part A, what you can say is, as the temperature increased, so did metabolism of the plant. And you can, instead of using metabolism, you could use oxygen consumption as well. And that would be okay because they basically mean the same thing in this case. So as the temperature increased, so did metabolism, or the rate of metabolism, or this oxygen concentration, or oxygen consumption of the plant also increased, because you want to actually mention that. So the metabolism of the plant also increased. It also asks you, so that's the first part, okay? That's just 1 point out of this question. The second thing it's going to say is you also need to discuss the effect of temperature on metabolism for each variety of seedling. So, according to the graph, variety B has a higher rate of consumption of oxygen compared to variety A. And if we look on our graph, here's variety B, and here's variety A. Okay, so we can see that B is much higher than A. So that tells us that B's oxygen consumption is much greater. Unfortunately, we're not done. So the reason for the increase in metabolism, we need to explain why. Because even though it doesn't really say, well, it does. It says explain the relationship, which means that you can't just give the relationship. That's part of it, but you also need to explain it. So in this case, the reason that there's an increase, so the reason for the increase in metabolism is because, and here's where things get interesting, because on this question, you can actually answer it in one of a couple of ways. And so when we talk about it, you could say that as temperature increases, the kinetic energy increases, which means that reactions happen faster. Or you could say that enzyme reaction rates increase with temperature. Or you could say that it could have an effect on the electron transport chain. Now, personally, I like the enzyme question, but if you can't remember it, you could just say that because an increase in temperature means an increase in kinetic energy, which speeds up reactions. Again, you could say, or you don't have to say all of them. You just have to come up with one of them, basically saying that, hey, when you increase temperature, this is what happens. And one of them could be an increase in kinetic energy, which would cause a reaction to speed up. Or the electron transport chain might speed up, or the enzyme reaction rate might speed up. Because you have to remember, in photosynthesis and respiration, almost all of these processes are controlled by a lot of different enzymes. So the higher the temperature, the more likely they are to run. However, if the temperature gets too high, enzymes are proteins, they will denature. So that's why photosynthesis is a very specific temperature range, which if you've watched the review video, will remember it's between like 0 degrees Celsius and usually 30 to 35 degrees Celsius, depending on the plant. Now for part B, in a second experiment, variety A seedlings at both temperatures were treated with a chemical that prevents NADH from being oxidized to NAD+. Predict the most likely effect of the chemical on metabolism and oxygen consumption on the treated seedlings. And then it asks you to explain your prediction. So for this one, we need to do two parts. The first part is predict the most likely effect of blocking NADH. Well, NADH is one of the hydrogen and electron carriers in respiration. So again, if you have a cheat sheet in front of you of respiration, you should remember that NADH carries protons and electrons. Specifically, it goes to the electron transport chain. So for part B, your prediction, you would expect if you have less energy going to the electron transport chain, you could expect that your metabolism either stops or slows down. Respiration could stop or slow down. So part B, the first thing I would say is that respiration could either slow down or stop. And then it asks you to explain why. Well, NADH provides electrons and hydrogen ions, or protons, to the electron transport chain. Without these two things, the ETC, or electron transport chain, you might want to write it out, the electron transport chain will stop producing ATP. And that's it. So the first part of the question asks for a whole bunch of stuff. So part A asks for a bunch of things, and then part B actually asks for quite a bit as well. Part B is 5 points out of the 9. So up here, they're looking for at least 4 things, and then down here, they're looking for 5. So if you don't get all of the points, you're still trying to aim for a specific number. It's not a big deal if you miss 1 or 2 points on a 9-point question. It seems like a lot, but it shouldn't be too bad if you're aiming for a 3. But you're trying to get as many points as possible without overgoing things or writing the wrong things down. So just keep that in mind. You don't want to start going on and on about photosynthesis when there's nothing about photosynthesis in this actual question. So just be aware of that. That's why NADH, recognizing that as respiration, versus NADPH, which is photosynthesis. FRQ 5 is a controlled experiment was conducted to analyze the effects of darkness and boiling on the photosynthetic rate of incubated chloroplast suspension. So we're going to boil some chloroplasts. The dye reduction technique was used. Each chloroplast suspension was mixed with DPIP, an electron acceptor that changes from blue to clear when it is reduced. Each sample was placed individually in a spectrophotometer, and the percent transmittance was recorded. The 3 samples were prepared as follows. Sample 1 is a chloroplast suspension plus the DPIP, which is just the chemical they use. Sample 2, the chloroplast suspension is surrounded by foil wrap to provide a dark environment, so no light. And you can totally write on these tests if you print one off, but that's going to take time. And then chloroplast suspension that has been boiled, so high heat was added, okay? It asks you to identify and explain the control or controls for this experiment. For question number 5, the first thing we want to do is we want to identify what the control is. A control is going to be your comparison group, so whichever group is the most normal group in this case. Unless it's called a negative control, then it's like nothing, okay? Like you're supposed to get zeros on those or no results. But in this case, the control is your normal group. So the normal group would be our light unboiled group. So sample 1 is our control group. So the control is sample 1. You also want to explain why. So sample one, let's see, sample one is treated under normal conditions with regular light. Regular amounts of light would be better. And normal temperatures. You might also want to explain that the control is the basis of comparison. By having a control, you're actually allowing for yourself to see if the experiment worked or not. Without a control, something to compare against, a lot of experiments just aren't worth anything because it could be a different factor going on. If you grow something under normal and then you do these other experiments and all the numbers end up really similar, there's no statistical difference, which would also mean that we would probably do our chi-square test if we were doing a prediction, or we might put our error bars in our graph to show that, hey, these two data points overlap. Now, in this case, they don't give that to us. Instead, they're just asking us, hey, what is the control? So in this case, we tell them this is what our control is. The other purpose of a control is it provides a reliability for your data or design. It shows that you're using identical procedures, reagents, measurements. As long as everything is the same, you should end up with the same kind of results. So that's FRQ5. FRQ6, if I recall correctly, is very similar to, or is the same as a test question that we saw earlier in the year. In this case, it asks us to predict the relative rate of photosynthesis in each of the three groups and then justify your predictions. So you could see something similar to this on the AP test. In this case, these are the two absorption spectra, and this is part of a much bigger question. There's like two other questions with a couple of points talking about bacteria. Specifically, this is a bacteria absorption spectra that also does something similar to photosynthesis, and these are the wavelengths they absorbed. And then this is a plant. This is chlorophyll A's wavelength absorption, okay? So if we look, we'll notice there's a peak here, the peak here, and then a major trough. There's like no absorption down here. And then we can look at our wavelength guide, and purple, blue, and then cyan and green are down here, and then yellow, and then orange, and then red are over here. And all it asks us to do is predict the relative rate of photosynthesis in each of the three groups, specifically looking at graph 2. Okay? And what they want is they want to know which one goes where. So we need to predict the relative rate of photosynthesis for each of these colors of light. That's what wavelength means. So our highest wavelength of our highest rate of photosynthesis would be expected to be in the 430 nanometers. And you would justify that because the shorter the wavelength, the more energy the light has, and it's absorbed more, which means more energy is being absorbed. So more energy means more photosynthesis can take place. So for this question, you would want to mention each individual wavelength of light, which one is highest, which would be the 430. The medium or intermediate would be the 650, and then the 550 would be the lowest. Highest amount of energy provided, medium amount of energy provided, very little energy provided, okay, which means more photosynthesis, some photosynthesis, very little photosynthesis. So that's what they're looking for. So that's your prediction, and then you would justify, hey, Y, which is how much energy is being provided for the actual reaction. So that's FRQ6. Again, I think this was on our test. FRQ7 is like a scenario. It's a 1-point question. This would have shown up on a regular AP test. I don't think it would show up now. So it's just a quick and easy question about, hey, what would happen if you had these two bacteria? Be careful because it talks about how toothpaste increases pH, and it talks about how some of them can't handle acidic, specifically S. sanguinis, can't handle acidic environments. But increasing the pH causes it to be basic. So you would expect to see S. mutans to probably die off because it's being exposed to oxygen, whereas S. sanguinis would probably thrive because it can handle the oxygen, and the pH isn't really bad for it. Question 8 is a really nice question for studying for the AP test. The pyruvate dehydrogenase complex, or PDC, catalyzes the conversion of pyruvate to acetyl-CoA, a substrate for the Krebs cycle. The rate of pyruvate conversion is greatly reduced in individuals with a PDC deficiency, which is a rare disorder. A is going to ask you to identify the cellular location where PDC is most active. Well, if PDC's job is to turn pyruvate into acetyl-CoA, we have to remember that pyruvate is the product of glycolysis, and acetyl-CoA is the first reactant of the citric acid cycle, or Krebs cycle. So part A asks us to identify, hey, where does this take place? Well, where does turning pyruvate into acetyl-CoA happen? Well, glycolysis is in the cytoplasm, but acetyl-CoA gets created in the mitochondria. So part A simply wants to know that this PDC happens in the mitochondria, specifically the mitochondrial matrix. So, but you only need to mention that it happens in the mitochondria. So PDC is found in the mitochondria. B asks you to make a claim. So our first part was identify, which is usually like a one thing, like, hey, tell me what it is. B asks you to make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount of NADH produced by the Krebs cycle in a cell. And then it asks you to provide reasoning to support your claims based on the position of PDC catalyzed reaction in the sequence of the cellular respiration pathway. If you have a nice cheat sheet that shows you what goes into and out of each step, you'll notice that PDC isn't going to affect NADH at all from glycolysis. So you would expect to find the same amount of NADH coming out of glycolysis because PDC is not needed. So the amount of NADH with, so the NADH from glycolysis would be the same with or without PDC because it is not needed. During glycolysis. Glycolysis has already happened. Okay, PDC turns pyruvate into acetyl-CoA. Now, you also need to talk about what happens to the NADH in the Krebs cycle. You would expect the Krebs cycle, the amount of NADH so the amount of NADH to come out of the Krebs cycle to would decrease because there is no acetyl-CoA feeding into the Krebs cycle. basically the acetyl-CoA is what goes into the Krebs cycle to make it run. So you would expect it to either slow way down or to stop completely. And then you also should mention that the Krebs cycle happens after pyruvate is turned into acetyl-CoA. The Krebs cycle occurs after pyruvate has been turned into acetyl-CoA. For part C, it says the PDC deficiency is caused by mutations in the PDHA1 gene, which is located on the X chromosome. A male with PDC deficiency and a homozygous female with no family history of PDC deficiency have a male offspring. Calculate the probability that the male offspring will have PDC deficiency. Whoa, that's a genetics problem. So C asks us to create a genetics problem. Now it's X-linked, which tells us sex-linked. Dad has it, so let's just go with X, X little p, Y. You just pick a letter. If little p doesn't work for you, pick a different letter. Mom is normal, so she's X big P, and then we just run through our Punnett square real quick. And what we find is they want to know what's the probability of a male offspring having it. The probability would be 0. Unless they ask you for like proof or explain why, you don't really need the Punnett square. Because if you remember, if dad has it and mom doesn't have it at all, their sons can't have it. Nobody will have it, in fact. If mom is homozygous purebred for something and it's sex-linked, none of their kids are going to end up with it. Okay? And so that's how you answer FRQ 8. We can ignore FRQ 9 because they are not going to ask you to do any sort of graphing on the AP test this year. Thank goodness. FRQ 10 is pretty complicated. It's got this graph and it's got a whole bunch of information that you have to read about, and then you have to figure out like, whoa, that's a huge word. But it's not as scary as it looks. The first thing you want to notice is that it's, we sometimes want to skip ahead and just read what the question wants. And it's actually on the next page. It says provide supports using data from figure 1 for each of the researchers' claims. And so we want to provide support for the 4 claims that are down here at the bottom. So the researchers made the following claims about the effect of KAR and the effect of TMB on seed germination relative to the control treatment. And you'll notice there's no mention of water. Water is actually the second part of the experiment, and we don't need that information at all, at least not for this part of the question. Instead, we just look at it and say, okay, KAR alone affects the timing of seed germination compared to the control treatment. Well, we look at our graph. Here's our control. And what we see is this is when the control starts to get, when germination starts to take place. So it starts to pick up here. And we notice that germination, for the most part with KAR, happens here. So in this case, germination starts sooner with the KAR than it does with the control group. And then if we look at percentage-wise, we'll notice that our percentages are higher with KAR than they are compared to the control group. The third claim says TMB alone affects timing of seed germination. Well, it does. TMB causes it to be a little bit later or slower, just a little bit. And then lower percentage of seed germination, well, yeah, TMB is way down here compared to our control group. We're just comparing. So in this case, you would have 4 sentences for each one worth of points. The last FRQ question is also a general information question. So it's probably not going to show up on the AP test. However, it is important to note that in these sort of problems, instead of seeing this and being asked, hey, describe one contribution of each of the following in ATP synthesis, which is just asking, hey, how does this create ATP or do something to help with creating ATP? Same thing with here and here, which by the way, this is all just respiration, glycolysis, Krebs cycle, etc. Instead, what they might do is they might screw one of these things up. They might say, hey, what were to happen if this protein was removed or blocked? Okay. Or, hey, what happens if this protein is messed up? What happens if the hydrogens never get across? What happens if acetyl-CoA never goes in? These are the kind of questions that are going to show up on the actual AP test. So just be aware with the energy questions that it's a really good idea to make cheat sheets for both photosynthesis and respiration. What I'm thinking of doing is at some point, I'd like to make one cheat sheet for you guys to look at, and then you guys can make them however you want, really. But it's a really good idea to have at least one per unit, if not one per major subject area. So it might be a good idea to have one for photosynthesis and one for respiration, with a bit of notes at the bottom about enzymes, because they're also part of this unit in AP biology. So this is the end of the FRQ review section. I posted both the questions and the key on Google Classroom. You can also expect to find a practice test posted on the AP Classroom in the next day or two, because they just posted a new practice test, and I'd like to toss one on there for you guys to look at. Now, I know AP Classroom is like the worst format in the world, but hopefully you guys will be able to use it and kind of get a feel for how the actual test is going to look. If you guys do have any questions about FRQs or cell energy, please feel free to contact me via email, and we can have a quick Zoom meeting, or if it's something I can answer over email, I'd be more than happy to. Otherwise, this is the end of the FRQ review section. I hope all of you have a good rest of your day.